Thanks for giving me the opportunity to present the work that we're doing at OrganoTherapeutics to break the wall of Parkinson's disease. It is indeed a tough wall to break, since more than 200 years have passed since the description of the pathology, and we still don't have any treatment that cures or stops the progression of the disease. As of today, it's a disease that affects more than 6 million people globally, and it is expected to double in the next 20 years. One of the major risk factors for contracting the disease is aging, and in the context of the future global demographics, the trend of increasing Parkinson cases is expected to accelerate. This scenario, combined with a reduced number of existing drug candidates, makes it an even more difficult task. We believe that one of the main reasons for this failure is that in the previous decades, modeling in the lab the loss of dopaminergic neurons, what is normally happening in the midbrain of a Parkinson patient, was done with inappropriate tools. Drug candidates were selected based on animal experiments or cultural cells flat in a dish. These two approaches have limitation in recapitulating the complex 3D architecture of a human brain. At OrganoTherapeutics, we believe that we have the necessary tool to tackle the disease, and this is the midbrain organoid, or mini-brain. We spent the last 6 years optimizing the model that starts with taking a skin sample from the patient, from where we isolate a type of cell called fibroblast. We reprogram this cell into a pluripotent stem cell, a cell that has the capacity of forming different cell types. From this point, we can guide them into becoming the cell type that we like in a process called differentiation. And we do so first into a neural stem cell, which are the ones that we in fact use to generate the midbrain organoids. These mini-brains can be maintained in culture for a long period of time in specific conditions that we streamlined over the past years. These organoids not only present the specific type of neurons that are lost in Parkinson's disease, they also present glial cells such as astrocytes and oligodendrocytes. And they're also functional by forming synapses, producing dopamine over time, and presenting electrophysiological activity and showing deposits of neuromelanin. Our solution is not only based in the model that I've just described for drug discovery, we are also adding 2 new strategies into the drug development pipeline. One of them is phenotypic screening, and this is allowing us to identify the compounds that are reversing the alterations that we see in the organoids from the patients. The other one is stratified screening, because we are using different mini-brains generated from patients that are having different genetic backgrounds. These strategies combined leads to a pipeline that accelerates the identification of more robust candidates, and these candidates don't follow the traditional one-size-fits-all, since we test our compounds in a stratified library of Parkinson patient-derived mini-brains. As an example of one of the 7 candidates in our pipeline, we see in this case a significant increase of the neurons lost in a Parkinson patient mini-brain after we perform a treatment with our lead compound. The next steps that we're aiming to perform at OrganoTherapeutics are preclinical testing and clinical trials of our lead compound, as well as pushing forward the other candidates that we have in the pipeline. We're also planning to screen more FDA-approved libraries to have more candidates in the future. We think that if we are able to achieve these steps, we will bring down the wall of Parkinson's disease. Thank you for your attention.